Hello everyone, this is Srushti Sonpasare and my topic is screening technique for new drug and new drug development. Topics covered will be drug development, clinical trials, phases of clinical trials. Development of hundreds of new drugs in the last few decades revolutionized the practice of medicine. Discovery and development of new drug is a time-consuming and expensive procedure. After identification, the structure of new compound and its purity are determined. If the compound is found to be promising, then it is subjected to preclinical evaluation in animals and clinical trials in humans. Preclinical evaluation. The compound is evaluated in animals for the following. First, toxicity studies include acute, subacute, and chronic toxicity studies, safety and efficacy evaluation, determination of therapeutic index. Third, pharmacokinetic studies. Clinical trials. When the drug is found to be reasonably safe in animals, it is subjected to clinical trials in human beings. Application is filed for investigational new drug IND status to test the drug in human beings. Clinical trials are conducted to know therapeutic efficacy and compare it with existing drug. Good clinical practice. Good clinical practice is an international ethical and scientific quality standard for designing, conducting, recording, and reporting clinical trials. The aim is to ensure that rights, safety, and well being of clinical trial subjects are protected. The clinical trial data generated is credible and genuine. Ethical clearance. After obtaining permission from the concerned regulatory authority to conduct the trial in a given setting, permission should be obtained from the local Institutional Ethics Committee IEC or Institutional Review Board IRB. The IEC looks into the ethical aspects of the trial and ensures that the trial is conducted ethically and the right of study participants are protected. Informed consent. For enrolling a subject into clinical trial, he or she should be informed in details about the trials including the risk involved and subject should willingly consent to participate in studies. This is to ensure that the participation in the study is purely voluntary and not by force. Clinical clearance after obtaining permission from the concerned regulatory authorities. Clinical trials are generally conducted in four phases, though phase 0 is also included in some situations. Phases of clinical trial. Phase 0, number of subjects 10 to 15 normal volunteers. Objectives explore pharmacokinetics and pharmacodynamics. Phase 1, 20 to 50 normal volunteers. Objective to establish safety, to know biological effects, pharmacokinetic profile, and to design a safe dose. Phase 2, number of volunteers 100 to 300 to establish efficacy, detect adverse effect, and pharmacokinetics. Phase 3, 250 to 1000 patients to establish efficacy, safety, to identify latent side effect, tolerance, design ideal dose range, and to compare with the existing drugs. Phase 4, more than 10,000 patients, long term safety and efficacy to identify other possible therapeutic uses. These phases are conducted by Phase 0, clinical pharmacologist, Phase 1, clinical pharmacologist, Phase 2, clinical pharmacologist and clinical investigators. Phase 3, clinical investigators. Phase 4, medical practitioner. Clinical trial registry. In order to make the drug development transparent, accountable, and data accessible clinical trial registry of India has been set up. Though the registration is voluntary and free of cost, it is required for the publication of data from the clinical trials. The details registered are freely accessible to all, including general public. Thank you.